Hello everyone. In a previous video we have studied about the microsporogenesis. Now we have seen the 4 stages separately. Let's see all the 4 stages together. So first we have the Microsporangium, which is smaller, which consists of the densely arranged sporogenous cells called as the archisporial cells. As the anther matures, the Microsporangium increase in size and the archisporial cells form space between them. Now this Microsporangium is called as the pollen sac and the archisporial cells which form space between them is called as the pollen mother cell, or in other words it is called as the microspore mother cell. The microspore mother cell thus formed is diploid in nature. This microspore mother cell undergo one meiotic division to give rise to the microspore tetrads, where the microspore tetrads or all the 4 microspores are haploid in nature. The microspores are kept together with the help of the callose thickening. The tapetal cell releases the callase enzyme. This callase enzyme dissolves the callose thickening. Now the microspores are formed individually. Now let's see about the formation of the male gametophyte. Now this microspore consists of a plasma membrane. The outer membrane is called as the exine. This outer membrane is called as the exine and the inner membrane is called as the intine. Now this structure is called as the pollen grain. The pollen grain is also haploid in nature. That is, the pollen grain consists of the plasma membrane. Inside the plasma membrane we have the intine. The intine is made up of cellulose and pectin, and outside the plasma membrane we have exine. The exine is made up of the sporopollenin. We have also studied that the tapetal cell releases sporopollenin, that is, it is a Yubish bodies, and this Yubish bodies are called as sporopollenin, the most resistant substance now till date. This sporopollenin are absent in certain places, that is, exine is absent in certain places, and this is called as the germ pore. Okay, we have 2 types. One, if it is circular germ pore, we call it as germ pores. If it is cylindrical in nature, we call it as germ furrows. We will see the role of these germ pores or germ furrows little later. So, in certain places, the exine is absent, that is, the sporopollenin is absent, and these places are called as the germ pore. Now, this pollen grain, which is haploid in nature, undergo one mitotic division. We know that in mitotic division, the karyokinesis takes place, followed by the cytokinesis. The karyokinesis is nothing but the nuclear division, where the nucleus divides into 2. Once the karyokinesis takes place, the cytokinesis takes place. In the cytokinesis, we can see the formation of 2 irregular cytoplasmic content. The one with the dense cytoplasm, this dense cytoplasm is called as the vegetative cell, or it is called as a tube cell, which consists of a prominent nucleus. Now, this larger cell is called as the vegetative cell because it does not take place any role in the formation of the male gamete. It is called as the tube cell because here the plasma membrane of this vegetative cell gives rise to the pollen tube. This pollen tube makes way through the germ pore. This is the main function of the germ pore. Germ pore play a very important role in making the way for the pollen tube. Okay. As the cell grows, now the smaller cell which is thus formed, it consists of the callose wall. This cell, smaller cell, floats in the larger vegetative cell. Now this small cell is called as the generative cell, and this cell is capable of producing the male gametes. Now recalling everything together, we have the Microsporangium, that is, the Microsporangium consists of the archisporial cells. This archisporial cells gives rise to the pollen mother cell. The pollen mother cell is diploid in nature. This pollen mother cell undergo one meiotic division to give rise to the microspore tetrads. The microspore tetrads thus formed are haploid in nature. Now the microspore tetrads are dissolved with the help of the callase enzyme to give rise to the individual microspore. When the microspore consists of the plasma membrane, the outer exine and the inner intine, we call this as the pollen grain. The pollen grain is also haploid in nature. In certain places, the exine is absent, and these areas are called as the germ pores, from which the pollen tubes moves out. Now, this exine, the exine mainly consists of sporopollenin, the most resistant substance on the earth. Now, why is this exine much harder? And why is this germ pore present? The exine is much harder because it consists of sporopollenin, the most resistant organic substance on the earth. Now we know that by temperature or by any hard enzyme or by acid base, the sporopollenin cannot be dissolved. So when the insect, that is the pollinators, feed these pollen grains, the sporopollenin will not be dissolved and it is intact. So if there is a presence of germ pore, the pollen tube formation takes place. Otherwise, the pollen tube formation does not take place. So every pollen grain will have germ pores through which the pollen tube germinates. Now, this pollen tube, the pollen grain, which is haploid in nature, undergo one mitotic division to give rise to the vegetative cells and the generative cell, where first the karyokinesis takes place, where the nuclear division takes place, then the karyokinesis is followed by the cytokinesis. The cytokinesis, the vegetative cell and the generative cell forms. The generative cell floats inside the dense cytoplasm of the vegetative cell. Now we have the mature pollen grains. Now we have the archisporial cells. The archisporial cells get mature to form the microspore mother cells, or instead it is called the pollen mother cell. The pollen mother cell undergo meiotic division to give rise to the microspore tetrads. This microspore tetrads forms the outer exine layer and the inner intine layer to form the pollen grains. Now these pollen grains have the vegetative cell and the generative cell. Once this is formed, the pollen grain has to be released out. Now how it is being released out? The epidermis, the endothecium and the middle layer, which together constitute the anther wall, ruptures. When this wall becomes rigid and fibrous, once this wall becomes rigid, this is the place where still these layers are not fibrous, and this causes the dehiscence of the anther, when this wall breaks out to release the pollen grains. This is how the pollen grain is released out, and this process is called as the dehiscence of the anther. Let's see about development of the male gametophyte. The male gametophyte development, that is the pollination, occurs in two stages. One is in the 2-cell stage, the other one is the 3-cell stage. In 60% of the angiosperms, the pollination occurs in the 2-cell stage, that is, it consists of a tube cell and a generative cell. Now this generative cell divides upon reaching the stigma, that is the male reproductive unit of the flower. Here the generative cell divides to give rise to the male gamete. The 3-cell stage, the 40% of the angiosperms undergo pollination in the 3-cell stage, where it consists of the tube cell and 2 male gametes. Inside the anther, the 2 male gametes are formed. So this is mainly seen in the cereals. Let's study about the features of pollen grains. The first important thing is the study of pollen grain. The study of pollen is called as the palynology. Next we have about the pollen products. In Western countries, these pollen products are used in the form of tablets and syrup. It is believed to increase the speed of athletes and the race horses. And the pollen products are also used in creams. It is believed that the pollen grains protect themselves from UV rays and the pollen creams protect the skin. Next feature about the pollen is the pollen viability. The period till which the pollen remains functional is called as the pollen viability. In the wheat and the rice, the pollen remains viable for 30 minutes. In few plants, in the few flowers, the pollen remains viable for few months. And this pollen is cryopreserved in the liquid nitrogen for about -196 degrees Celsius. Next about the pollen allergy. So many people are allergic to the pollens. and this pollen allergy causes the hay fever, and this is mainly because of the Parthenium that is called as the carrot grass. This carrot grass or the Parthenium is introduced into India as a weed along with the imported wheat. Now this pollen allergy also causes the respiratory illness such as the asthma and bronchitis. In this video, we have studied about the formation of the male gametophyte, that is the pollen grain, and we have also studied about the development of the male gametophyte in the 2-cell stage and the 3-cell stage. We have also studied about the dehiscence of the anther and the features of the pollen grain. In our next video, let's study about the female reproductive unit and the female gametophyte. Until then, happy learning from Edge to all.